Hello everyone, welcome to the Infectious World channel. Today I will be talking about TB vaccines. The BCG vaccine, which is used to prevent TB, has been in use since 1921. It used worldwide. It was developed by French researchers Albert Calmette and Camille Goram. They accomplished this by passaging M. bovis in vitro for almost 13 years. BCG's effectiveness against adult TB varies significantly, from no effect to up to 80% effectiveness. But why is that? Well, the two most relevant hypotheses for explaining BCG's ineffectiveness against pulmonary TB in adults are the masking and blocking hypotheses. Understanding the masking hypothesis. The masking hypothesis proposes that immunization with environmental mycobacteria, EMB, provides a significant level of defensive immunity against Mycobacterium tuberculosis, resulting in diminished effects of subsequent BCG vaccination. It is suggested that EMB hinders the effectiveness of BCG to provide protection against TB bacteria, despite its ability to offer some level of protective immunity against MTB. Now let us talk about the blocking hypothesis. The blocking hypothesis suggests that prior exposure to EMB triggers powerful antimycobacterial immune responses that hinder BCG replication, thus negating its effectiveness in generating protective immunity against subsequent MTB infection. Let us now explore tuberculosis in infants and young children. It is worth noting that infants and young children may not be able to successfully contain the MTB bacteria. In young children, CD4 plus T cells are recent thymic emigrants with defective functionality and differentiation and are biased towards Th2 effector cells. The immunological environment in young children studies have demonstrated that the immunological milieu in young children is inclined towards the Th2 aspect, thereby suppressing the Th1 type of immunity and inflammatory pathways. Furthermore, young children's CD4 plus T cells are newly produced by the thymus and have impaired functionality and a preference for Th2 effector cells. Enhancing the immune system in children with BCG vaccination, as I have already alluded, research shows that children have an underdeveloped immune system, resulting in weakened Th1 responses and inflammatory pathways. Additionally, infant CD4 plus T cells demonstrate suboptimal functionality and differentiation bias towards Th2 effector cells. However, BCG vaccination has been shown to improve the immune profile and induce MTB-specific Th1 polarized immune responses in children. Interferon gamma, IFN-GUNG, is produced by CD4 plus T lymphocytes in BCG-vaccinated infants. BCG vaccination in neonates has been observed to stimulate cytotoxic T lymphocyte responses to Mycobacterium. In infants, the BCG vaccine elicits immune responses from CD4 plus and CD8 plus T cells, as well as the gamma delta T cells. These immune responses are critical in the fight against Mycobacterium tuberculosis. BCG-vaccinated newborns have lower levels of Th2 cytokines compared to Th1 cytokines. Why is it important to administer BCG vaccination to newborn infants? Well, when given to newborns at birth or in early infancy, it has proven effective in preventing the spread of TB in young children. It has also been shown to be effective in preventing the spread of TB in young children. WHO's NTB strategy. WHO's NTB strategy seek to reduce global TB incidence by 90% and TB mortality by 95% by 2035. In order to achieve that, there must be a new vaccine that is effective across all age groups, particularly adults and adolescents. Vaccination is also the most effective way to manage the rapid spread of multi-drug-resistant tuberculosis. WHO called for development preferred product characteristics, PPC, for new TB vaccines. The first PPC involves development of a vaccine for adolescents and adults. Furthermore, the vaccine need to provide protection without evidence of latent MTB infection. The second set of PPCs involves development of vaccines to improve on BCG in infants. Now let's look at the WHO goal of developing a safe, effective, and affordable TB vaccine for adolescents and adults, which entails preventing MTB infection and TB disease in infants and young children, as well as in adolescent and adult contacts. This vaccine should be protective in those with or without evidence of MTB infection. It should also prevent progression to TB disease following primary infection, as well as following reinfections and reactivation in those with latent infection. Let us now take a look at the WHO's second preferred product characteristic: developing an affordable TB vaccine for neonates and infants with improved safety and efficacy as compared to BCG. Infants and young children with TB are not an important source of MTB transmission. However, they represent an important vulnerable group, and as such, a need arises to improve upon the current BCG vaccines. Clinical pipeline of new TB vaccines. Regarding the new TB vaccines, the following are the number of strategies for their application. 1. Preventive pre-exposure for uninfected individuals. 2. Preventive post-exposure for latent TB adolescents and adults. 3. Therapeutic for active TB. Now, let us briefly describe the new TB vaccine strategies. The pre-exposure vaccines aim to induce a more robust protective immune response than BCG in order to prevent the infection from being established. The post-exposure vaccine strategy aims to prevent disease reactivation by inducing a robust and long-lasting response. Another approach for categorizing vaccines in preclinical and clinical trials places vaccines in their biochemical forms: live attenuated, inactivated, adjuvanted protein subunit, and recombinant. Others are meant to boost the current BCG vaccine. Currently, there are at least 23 vaccine candidates in clinical trials in humans. They include live attenuated, inactivated, subunit, and recombinant vaccine types. Modified BCG vaccines, based on modified BCG, have demonstrated efficacy in Phase 3 clinical trials. These vaccines contain a gene encoding Listeriolysin O, LLO, from Listeria monocytogenes and the removal of the BCG urease C gene. Inactivated vaccines. The DAR901 vaccine is the inactivated vaccine. It was designed from inactivated Mycobacterium obuense. It was meant to be used in a heterologous vaccination approach, where adolescents who received BCG as infants were given DAR91 as a booster dose to enhance immune protection. However, this vaccine failed to protect BCG-vaccinated adolescents in Tanzania from TB occurrence in a Phase 2b clinical trial. Subunit TB vaccines H56, IC31, H4, IC31, and M72 ASR1 are TB subunit vaccines and have completed Phase 2 clinical trials and demonstrated effective immunogenicity and safety in human volunteers. Viral vectored vaccines. They are designed using whole cell vaccines and subunit vaccine approaches. The viral vector vaccines that have completed Phase 1 clinical trials are AU85A, AU35. Another viral vector vaccine, MVA85A, has recently completed a Phase 2 trial in South Africa. Inactivated TB vaccines. These vaccines induce both Th1 cell-mediated and humoral immune responses and have demonstrated good immunotherapeutic effects in controlling TB. Inactivated vaccines undergoing clinical trials currently include MIPIP, RUDI, VACE, and DAR901. Other inactivated whole cell vaccines which have completed Phase 2 trials successfully are RU2 and oral V7 vaccine, which have completed a Phase 3 clinical trial. Attenuated TB vaccines. Attenuated TB vaccines are prepared by removing some of the virulence genes in the MTB. They can be used as a preventive vaccination and a substitute for newborn BCG vaccination. Attenuated vaccines are able to activate complex and diverse immune responses and have a diverse range of antigenic epitopes, which allows for induction immune responses similar to natural infection. MTB-VAC and BCG revaccination are attenuated TB vaccines in clinical trials currently. Recombinant BCG vaccines. Recombination and modification of existing BCG is currently researched. Different types of recombinant BCG or BCG have been developed and had their protective effects and humoral and cellular immune responses evaluated in animal models and humans. 3 types of RBCG have entered clinical trials: VPM2, RBCG30, and ERS422. However, RBCG30 and ERS422 were discontinued due to poor efficacy and safety issues. Thank you for watching. Please don't forget to like and subscribe.